Hello students, I welcome you to EPG Pathshala. My name is Dr. Shiv Kumar Sood. I am Senior Scientist in the Division of Animal Biochemistry at National Dairy Research Institute, Karnal in Haryana, India. Today I am going to talk about the module Basic Local Alignment Search Tool, that is BLAST, from the paper Biostatistics and Bioinformatics. Basic Local Alignment Tool, BLAST, is an online similarity search tool which finds the sequences in the database having similarity in local regions to the input or so-called query sequence in the hand of users. It uses local sequence alignment between the input query and each of the target sequence in a selected database. BLAST may find one or more local subsequences in the query which shows similarity to a local subsequence domain in the target sequence. BLAST reports statistical significance and actual alignment, which is to be used to infer homology. Since homologous sequences have similar structures and consequently similar function, a newly determined sequence can be used to find similar sequences in the database to infer structural, functional, and evolutionary relationship with the known sequences in the database. In addition, BLAST is used to identify members of the query gene or protein with the sequences in the database. A functionally interesting protein, say a spot in two-dimensional gel electrophoresis, can be sequenced using mass spectroscopy and used as query sequence for searching similar sequences in the database having, if known and structural functional information, then if this structural and functional information or knowledge is known for some similar protein in the database, then the same knowledge may be applied in understanding the unknown structure and unknown function of this query protein, which is, let us say, differentially expressed in two samples, say a normal sample and a diseased sample of the cell culture. With this goal, students will use BlastP specifying required input parameters to search known homologous proteins in the database. In this module, the students will understand BLAST for searching similar protein and nucleic acid sequences from the databases. A heuristic approach to reduce the number of target sequences for search as implemented in BLAST will also be explained. Input parameters for the BLAST variants, that is BLastP, BlastN, BlastX, tBlasN, tBlasX, available for gene discovery will be introduced. Finally, we'll have a look at Genome BLAST for searching assembled genomes and specialized BLAST for specialized applications in biochemistry, molecular biology, and biotechnology. BLAST implements a heuristic approach which results in substantial computational savings during similarity search. In this approach, the database to be searched is first filtered using the query sequence in a very fast manner to exclude all dissimilar sequences. This reduces the number of target sequences to be evaluated using dynamic programming alignment to a manageable number. However, actual implementation is different from the one implemented in FASTA. During initial filtering, the word list is extended to include all the words with allowed mutations to detect similarities. For matching diagonals, counting of score is using the given matrix. The diagonals are not joined but extended in both directions. Dot plot diagonals having at least two matching words are extended on both sides of the word without gaps till the score to give the highest segment pair called an HSP falls below a threshold. HSP is then aligned using dynamic programming, allowing extension to introduce gaps till the alignment score is above certain threshold and are reported as local alignments. Consequently, more than one alignment may result for one query sequence. An alignment between two sequences having identical and similar residues usually includes short diagonals similar to dot plot diagonals. Therefore, when a user submits a BLAST search using a query sequence, as in this example, glutamine, leucine, asparagine, phenylalanine, serine, alanine, glycine followed by tryptophan, BLAST splits this query sequence into overlapping words of specified size. The size specified is 2 in the present example. Therefore, all overlapping words of the size 2 are created. This is followed by introduction of possible mutations in each original word and scoring each mutated word using a substitution matrix PAM 120 to keep mutated words with a score above pre-computed threshold for the original word. This is called extended list of original words to allow detection of both identical and similar residues. The extended list is also known as lookup table. All the words in the list are called seeds for searching database. This is followed by looking up database sequences for matching words in the extended list using a given scoring matrix and keeping sequences containing matching words with recording offset which represents diagonal in dot plot. This is followed by extending alignment of the matched word in both directions by aligning identical and similar residues. By aligning identical and similar residues in two sequences. Extension requires two matching words on the same diagonal. Extension continues without introducing any gap till the score of the alignment in both the directions is above a prefixed threshold to result in a contiguously aligned segment known as high scoring segment pair (HSP). BLAST chooses highest scored HSP for alignment using restricted Smith-Waterman dynamic programming alignment on a centered dipeptide in highest scoring 11 amino acid long HSP. BLAST allows gaps to be introduced. Therefore, the highest scored HSP chosen for alignment using dynamic programming allows extension of the alignment in both the directions to introduce gaps till the alignment score is above a certain threshold. Dynamic programming is followed by statistical calculations for bit scores and e-volume for use in evaluating the statistical significance of each alignment generated during search. There are 5 BLAST variants available for similarity search. To retrieve a specific target, user has to choose a BLAST program to algorithm to use. Further, BlastN and BlastP has different programs to select. Nucleotide BLAST is selected to seek identical DNA sequences and splicing patterns. DNA query is used to search a DNA database, and the comparison between the sequences takes place at DNA level. Protein BLAST is selected to find homologous proteins. Protein query is used to search a protein database, and comparison takes place at protein level. BlastX is selected to analyze new DNA to find genes and seek homologous proteins. DNA query is used to search a protein database, and the comparison is at protein level. tBlasN is selected to search for genes in unannotated DNA databases. Protein query is used to search a DNA database, and comparison is at protein level. tBlasX is selected to discover gene structure. This search is translated nucleotide database using a translated nucleotide query. DNA query is used to search a DNA database, and the comparison takes place at protein level. Keeping in mind the target to achieve, formulation of a similarity search experiment with BLAST begins with a decision about what type of sequences to compare: DNA, protein, or DNA as protein. If the sequence under consideration is a protein or a gene encoding a protein, then search should probably take place at the protein level. Therefore, the database to be searched is decided, and database size can be limited through an entree query provided on the input section of the BLAST interface. To target search in a particular organism is also allowed. In alignment parameters, word size is decided to achieve sensitivity, desired sensitivity of search. Then, depending upon the homology targeted, a substitution matrix is selected for scoring the alignment. Keeping in mind the homology targeted, gap penalty costs are also fixed. The pre-treatment to be given to a query sequence, if any, and to the database sequence, such as filtering subsequences for low composition, is also decided. Then the user had to decide the output parameters, such as statistical threshold to include significant matches and number of sequences to be reported. Once the output is produced, user has to interpret the output alignments to draw an inference of homology. All the 5 BLAST variants are available on the same page as separate tabs. User can open any tab to use specific BLAST. Translated BLAST, that is BlastX tab, is opened in the present example. When the query is DNA, BlastX and tBlasX requires to choose a genetic code for translation of the queried DNA sequence, because protein databases are searched by these programs to analyze new DNA for genes and to discover gene structure. Now open the BlastP tab and paste the protein sequence of Glycophorin A from human in the sequence input text box. While setting search set for BlastP and Blastx, there is an option to exclude a particular organism. This will allow search of orthologous and paralogous sequences. To exclude an organism, checkbox is checked. This search will return only orthologous sequences from the database. If the checkbox is not checked, then the search will return only paralogous sequences. There is an option for entree query to report results for given entree keywords. Use entree query syntax to search a subset of the selected BLAST database. This can be helpful to limit searches to molecule types, sequence lengths, or to exclude organisms. Open the drop-down list for selecting a database and select UniProt KB SwissProt database. If one is interested in searching for proteins of known structures, then it is best to use just PDB database. The non-redundant protein database combines data from non-redundant GenBank CDS translations, PDB, SwissProt, PIR, and PRF. This removes the redundant, that is, superfluous identical sequences, and yields a collection with nearly all known proteins without repeating a given sequence. So, in the NR database, when they are when the sequences are taken from two different databases, same sequence is just included once only. That is called non-redundant database. BlastN and BlastP provides different programs for conducting similarity search for standard nucleotide and standard protein searches. For standard nucleotide BLAST, user can choose out of MegaBLAST, discontiguous MegaBLAST, or BlastN. The MegaBLAST is intended for comparing a query to closely related sequences and works best if the target percent identity is 95% or more and is very fast with word size chosen. It is available for word size range from 16 to 256 options. Discontiguous MegaBLAST uses an initial seed that ignores some bases, that is, allowing mismatches, and is intended for cross-species comparison. It is available with word size 11 and 12. BlastN is slow but allows a word size down to 7 bases with 11 and 15 options also available. For standard protein BLAST, users can choose from BlastP, tBLAST, phyBLAST, and deltaBLAST. BlastP simply compares a protein sequence to a protein database with option of word size 2, 3, or 6. tBLAST allows the user to build a PSSM, that is position specific scoring matrix, using the results of the first BlastP run with the option for word size of 2 or 3. phyBLAST performs the search but limits alignments to those that match a pattern in the query with the option of word size 2 or 3. Finally, deltaBLAST constructs a PSSM using the results of the Conserved domain database search and searches a sequence database with option for word size of 2 or 3. One can BLAST at this stage or open algorithm parameters. BlastN and BlastP provides different programs for conducting similarity search for standard nucleotide and standard protein searches. For standard nucleotide BLAST, user can choose out of MegaBLAST, discontiguous MegaBLAST, or BlastN. The MegaBLAST is intended for comparing a query to closely related sequences and works best if the target percent identity is 95% or more and is very fast with word size chosen. It is available for word size range from 16 to 256 options. Discontiguous MegaBLAST uses an initial seed that ignores some bases, that is, allowing mismatches, and is intended for cross-species comparison. It is available with word size 11 and 12. BlastN is slow but allows a word size down to 7 bases with 11 and 15 options also available. For standard protein BLAST, users can choose from BlastP, tBLAST, phyBLAST, and deltaBLAST. BlastP simply compares a protein sequence to a protein database with option of word size 2, 3, or 6. tBLAST allows the user to build a PSSM, that is position specific scoring matrix, using the results of the first BlastP run with the option for word size of 2 or 3. phyBLAST performs the search but limits alignments to those that match a pattern in the query with the option of word size 2 or 3. Finally, deltaBLAST constructs a PSSM using the results of the Conserved domain database search and searches a sequence database with option for word size of 2 or 3. One can BLAST at this stage or open algorithm parameters. BLAST algorithm parameters has 3 sections: general parameters, scoring parameters, and filtering and masking sections. BlastP general parameters section provides for the selection of word size out of 2, 3, and 6. Higher the word size, the more sensitive the search will be. BLAST An algorithm general parameters section provides for selection of word size ranging from 16 to 256. Again, higher the word size, the more sensitive the search will be. Expect value setting specifies the statistical significance threshold for reporting matches against database sequences. The default value 10 means that 10 such matches are expected to be found merely by chance, according to the stochastic model of Carlin and Asher. If the statistical significance ascribed to a match is greater than the expect threshold, the match will not be reported. Lower expect thresholds, that is below 0.01, are more stringent, leading to fewer chance matches being reported. BLAST algorithm next section is for setting scoring parameters. BlastP, tBlasXN, and BlastX provides options for choosing scoring metrics, option for setting gap costs, as well as option for setting compositional adjustment. On the other hand, BlastN algorithm provides only match-mismatch scoring. Linear gap costs are available only with MegaBLAST and are determined by match-mismatch scores. Compositional adjustments are not allowed. BLAST algorithm last section provides for filtering low complexity regions. The statistics for database searches assume that unrelated sequences will look essentially random with respect to each other. However, certain patterns in sequences violate this rule. The most common exceptions are long runs of a small number of different residues. For example, a polyalanine tract. Such regions of sequences could spuriously obtain extremely high match scores during alignment. For this reason, the search program may automatically remove such sections in proteins by replacing them with unknown amino acids such as the symbol given X. tBlasX algorithm provides for choosing a scoring matrix only. Gap costs and compositional adjustments are not allowed. To infer homology between the query and the target, interpretation of BLAST search alignments is undertaken using statistics. The BLAST server reports two measures to assess the significance of alignment. These are bit score and e-value. The raw score for an alignment is calculated by summing the column score for each aligned position using a specified substitution matrix and the score for each gap position using affine gap cost. Raw score of the alignment is not reported by the BLAST. However, BLAST reports normalized scores for each alignment, and this is called bit score. Raw scores have little meaning without detailed knowledge of the scoring system used. That is, unless the scoring system is understood, citing raw score alone is like citing a distance without specifying their units, that is feet, meters, or light years. Bit score has a standard set of units, that is bits of information. E-value, that is expectation value, is defined as the number of different alignments with a given score equivalent to or better than this score that are expected to occur in a database search just by chance. Lower the e-value, more significant the alignment. The most significant alignment is indicated by highest bit score and lowest e-value. However, one should actually look at the quality of the alignment before accepting the score. BLAST interpretation of statistical parameters include interpretation of bit scores. BLAST normalizes the raw score of the alignment to bit score using the formula shown in the box. Parameters k and lambda can be thought of simply as natural scales for the alignment search space size and the scoring system respectively. And S is the raw score for the alignment achieved during dynamic programming using a specific scoring matrix and gap penalties. Higher the bit score, more significant is the alignment. Dear students, as biochemists, let us use BlastP to find structurally and functionally homologous sequences to human glycophorin A protein. In Enter Query Sequence section of BlastP, paste the amino acid sequence of the mature peptide in plain text and enter name for the job title as GLPA_human. In Choose Search Set section, select non-resultant protein sequence database and organism to set to Homo sapiens. The non-redundant means that a given protein sequence is not present twice in this database. NR database is compiled by NCBI from non-redundant GenBank CDS translations, PDB, SwissProt, PIR, and PRI, and PRF, excluding the environmental samples from WGS projects. Enter Homo sapiens with text ID 9606 and check Exclude checkbox to search only orthologs of glycophorin A in human. However, in present example, checkbox is not checked, but Homo sapiens text ID 9606 is entered. Therefore, the search will report only paralogs for the protein glycophorin A in Homo sapiens. In the algorithmic parameters section, enter expect threshold 0.01. Select the word size 6 to make the search most sensitive. Select Blossom 62 to search intermediately diverged sequences with intermediately gap opening penalty 11 and low extension penalty 1. This will search intermediately diverged sequences. The expect threshold setting specifies the statistical significance threshold for reporting the matches against database sequences. Clicking BLAST button will show the results in a separate window because the checkbox Show Results in a New Window is checked. The results page has 3 sections which are important: graphic summary, descriptions, and alignment. In addition, the hyperlinks for other reports, including search summary, taxonomic reports, distance tree of results, and multiple alignment is also presented. This first part of graphic summary presents that putative conserved domain has been detected. User can click on the image Glycophorin A superfamily link for detailed results on the conserved domains. Next part of the graphic summary presents distribution of BLAST hits aligned to the query sequence. The score of each alignment is indicated by one of the 5 different colors, which divides the range of the scores into 5 groups. Multiple alignments on the same database sequences are connected by striped colored lines. Mousing over a hit in this striped line sequence causes the definition and score to be shown in the window at the top text box. Clicking on a hit sequence takes the user to the associated alignment. For the time now, scroll down the results page to reach description section. The description section presents an ordered list of the matches according to the scores and e-values, with best hit listed on the top. click on the second entry, Glycophorin B. This will take you to the actual alignment. The alignment here shows that a local region in the query sequence of human Glycophorin A aligned with the Glycophorin B local region. To reach this entry, click on SP accession number P06028.3 hyperlink. The complete entry will be displayed. In this way, user can scan the relevant entry. In the descriptions section to extract functional information about this protein. Similarly, if three-dimensional structure of similar protein is available, user can use those structure for understanding the molecular mechanism of its functioning. BLAST Cobalt gives multiple alignments. Therefore, clicking on the multiple alignment hyperlink in the Other Reports tab on the top of the page will bring this multiple alignment. In addition to the multiple alignment, the results window presents ligands. The results window presents ligands for links to other resources, including Unigene, GEO, that is Gene Expression Omnibus, Gene Structure, and MapViewer. Fourth entry ends with S to indicate that structure is available. Ending with other symbols like G and M indicates external links to the Gene and MapViewer databases. This helps to draw an inference for the target information. SmartBLAST processes protein query to present a concise summary of the 3 best matches from the non-redundant protein sequence database, along with 2 best protein matches from well-studied reference species. If possible, the 2 matches from the reference species database will be from different organisms. SmartBLAST produces the results using a combination of an optimized BlastP, a new implementation of BLAST meant to find closely related matches, and a multiple alignment. Additionally, SmartBLAST presents conserved domain database matches to the query. Search with phosphatidyl glycerol lysyl transferase from Enterococcus faecalis shows 3 best matches from Enterococcus faecalis and 2 best protein matches from well-studied reference species, that is Bacillus subtilis and Pseudomonas aeruginosa. To see annotations of the graphical output, just browse. To see annotation in graphical output, just browse the mouse over the bar to see the annotation. In the BLAST assembled genomes page, start entering Homo sapiens. As you begin, the drop-down list will suggest available genomes. You can select from the suggested genomes. If the genome is not suggested, then you can click a broad category written below this text box. However, for the present example, start typing Homo sapiens and select the suggested option with text ID 9606 and click Go button. This tool searches similar nucleotide sequences from a chosen assembled genome. Consequently, this tool is useful for finding similar sequences in one genome. If the query sequence is also from the same assembled genome, then this is equivalent to searching paralogous sequences in the chosen assembled genome. Visit UCSC genome browser and search for human lactate dehydrogenase, which is a tetrameric enzyme with 2 paralogs. One paralog is exclusively expressed in heart and other in muscles. These are different forms of the same enzyme within a single organism that catalyzes the same reaction but differ in their Km and Vmax values, as well as regulatory properties. These different forms of paralogous enzymes catalyzing the same reaction are called isozyme or isoenzymes. In the results page, click hyperlink for the first entry in the result list to display DNA sequence in browser window. Select all the text to copy in the displayed window. Paste the DNA sequence of human lactate dehydrogenase chain A in BLAST assembled genomes and click BLAST button. The results page shows the paralogs. The sequences with e-values more than 0.01 are significant to be true paralogs. Evaluate them statistically as well as have a look at the real alignments. Specialized BLAST at the home page of BLAST provides several BLAST variants for conducting specialized experiments using BLAST. For example, to make specific primers, use primer BLAST. All these BLAST variants are self-explanatory, so try and use them. Dear students, in this module, we learned BLAST tool for searching similar sequences in the databases. Since homologous sequences have similar structure and consequently similar function, a newly determined sequence can be used to find similar sequences to discover orthologs and paralogs of the sequence in hand. This homology search may be conducted to infer structural, functional, and evolutionary relationship with the known sequences in the database. In addition, BLAST may be used to identify members of the family of the queried gene or protein. A heuristic approach to reduce the number of target sequences for search as implemented in BLAST was also explained. The input parameters for BLAST variants, that is BlastP, BlastN, BlastX, tBlasN, and tBlasX, available for specialized gene discovery, were also explained. The interpretation of the similarity search results to infer homology was presented, and then SmartBLAST processes protein inquiry to present a concise summary of 3 best matches from the non-redundant protein sequence database, along with 2 best protein matches from well-studied reference species. Then we had a look at the genome BLAST for searching paralogs and specialized BLAST for specific applications in biochemistry and molecular biology as well as biotechnology. I thank you all for visiting EPG Pathshala.